All right. Our next speaker, Daisuke Kihara from Purdue, is going to give us an introduction to their methods development in structural modeling, validation, and refinement assisted by deep learning. Thank you, Daisuke. Take it away.
All right. Thank you very much for the opportunity. So today I have my lab member, Genki Terashi, who is now on the video. So the plan is that I spend about the first 20 minutes to overview our tools and how it works and some performance evaluation. And then I pass this to Genki, and then he will show practically, which way, how to use those tools. We have web servers, and also all the tools are downloadable, and he will talk about those practical aspects. So first, probably I can just quickly comment on the EM GAN. Mark did a really great summary of how to use these AI-based tools. And I can specifically comment on our EM GAN. EM GAN is trained on protein structure only. So we did not evaluate how it performs on ligands, or or even DNA or RNA or glycans. So please be very careful if you know that your map has those molecules other than proteins. For proteins, it will enhance the side chain, main chains very well, and which makes it easier to do the protein structure modeling. But it's not intended to identify unseen difficult ligands which are difficult to see in the map. And it's all, well, I was a bit surprised to see that, good to see that him is also sharpened by the EM GAN, but it is not in the training set. All right, so okay, so then I go to my presentation. So all right, so can you see the screen? It's okay. All right. So
Yep.
we have been using developing the AI-based deep learning-based tool. Oh, thank you. So and first a series of them, and we have tools for relatively higher resolution and lower resolution, because, well, as probably everyone knows here, the resolution, the information you have in the map really depends on the resolution. Here, I picked up just one random structure EM map from at 1.92 angstrom. There you can see the side chains also very well. When it goes to 3 angstrom, then the side chains details are lost for many cases, and even the main chain often lost in low resolution regions, local regions. And then when it goes to 6 angstrom, beyond 5 or 8, then, well, you can still see the secondary structures for most of the cases, but the main chains are even difficult to trace. And then, well, if it just goes over 10 angstrom, then you could see the shape, but the details of the structures, main chain, are completely lost. So we have been developing tools for specifically for certain range of the resolution. So here I show you the like chart of the resolution, global resolution. So if you use deep learning, well, if you use deep learning up to around 5 angstrom, most of the cases we can detect C alpha position, and main chain, we can do the main chain trace. And side chains are detectable by deep learning. And so we can assign the sequence along the main chain. And then ligand is also detectable. So by applying deep learning, we gain about 1 to 2 angstrom. In, I think at 2 to 3 angstrom, you can do the modeling without much problem using the conventional tools. But if you use deep learning based tool, you can gain about 2 angstrom and then facilitate the modeling. Well, at the 10 up to 10 angstrom, you can see the secondary structures. Now, okay, this is a tool you could use up to 2 to 3 angstrom. You can use conventional tools, and then up to about the resolution around 3 to 5 angstrom, the deep learning based tool we developed a deep MainMust for protein modeling and CryoRead for DNA and RNA. These tools can help you to do the modeling. And I will also show you the validation score called DAQ. And DAQ will use a deep learning network to identify, to verify if the amino acid you placed in each position agrees with local density patterns. Now at up to 10 angstrom, yeah, we can clearly see the secondary structure pattern by deep learning methods. And then we also have, now we have the structure fitting methods up to beyond 5 angstrom. The tracing main chain are difficult, even using deep learning, but we can do the structure fitting by using deep learning better than just against the low resolution CryoEM map. So I go through those couple of key programs from the next slides. So the first methods I'm going to introduce is deep MainMust. This is a protein main chain tracing modeling methods up to about 5 angstrom. Beyond 5 angstrom, this method has difficulty to do the modeling because it's too low. Even a deep learning cannot see the amino acid positions. An idea of deep MainMust is very straightforward. It is actually mimicking, following how you would do the modeling. So at the very beginning, you have a CryoEM map of up to about 5 angstrom. Then at the very beginning, we use deep learning network. This is the architecture called UNet. And this by using this deep learning, we identify the potential C alpha positions and also the other atom positions. And at each C alpha position, we also predict probability of 20 different amino acids type. So now I know where the atoms locate, C alpha locates, and the amino acid types locate. Then using that, the subsequent steps are really just connecting these dots. And then, okay, so we connect these dots by using so-called vehicle routing solver. This is, well, I don't have time to go into detail, but this is really connecting those neighboring C alpha positions and then make a fragment. When the resolution is relatively high, like a 2 to 3 angstrom, then this method may be able to connect all of the amino acids all together into the whole protein sequence. But if it doesn't happen so often, when the resolution is around 3 to 4 or going up to 5, then I cannot connect all of the C alpha positions, but I have lots of fragments. Some fragment connections might be wrong, but at least I have a library of fragments connected by built by connecting those dots. Now, to each of these fragment C alpha positions, using the amino acid types predicted, I assign the protein sequence. So now this fragment also have the sequence assignment. Well, and up to here, well, this library contains a correct fragments and the correct sequence assignment, and also inevitably, well, there are wrong ones. But now we want to connect those fragments together so that it will follow the sequence pattern. So this will be probably very similar to how you model the structure manually. Now, okay, so this is coming from the direct tracing. We also use AlphaFold, or you can use your own structure. Well, because AlphaFold does a very good job in the modeling, but probably you notice that when you use AlphaFold 2 or 3, you notice that sometimes an AlphaFold gives a very good fragments or domain, but entire structure is not right for your specific CryoEM map. So what we do is that although we use AlphaFold to model, we only use fragments only when they fit at least in the majority of the residues at the good at the density. So now we have fragments coming from direct tracing on the left, and then fragments coming from AlphaFold, and then library is composed of these two types of fragments, and then those are assembled together to build a structure. So that's an overall idea. I think it's probably very intuitive. Now, okay, so I'll show you a couple of examples of when the tracing works and when AlphaFold 2 works. In this example, this is this as you can see, this is a protein of two domains: alpha helical domain on the left and the beta domain on the right. And in this, okay, this is a 2.6 angstrom resolution. At this resolution, deep main must base. Base is just a tracing without AlphaFold. Can trace the CL main chain really well. RMSD 0.6 angstrom. But if you run AlphaFold for this protein, two domains are correct, but the orientation of the two domains are not right. I think it's a typical errors of AlphaFold. So if you just compute RMSD of this AlphaFold, well, it goes to 10 angstrom because only one structure is, one domain can be aligned well because orientation is wrong. And if we combine these two, we gain a little bit. In this case, tracing is working really well. This is the opposite scenario when the tracing is not working very well and AlphaFold help is critical. So this is 4.1 angstrom resolution. At this resolution, some part of so the pink chain in pink is PDB, and then thin lines are our main chain tracing. So you see that some part like this, the right bottom corner, are not traced very well because the local resolution at this position is low. So if we compute the TM score, this is kind of percentage of amino acids which are correctly aligned. It's about 66%. But AlphaFold doesn't care about the density at all, and they make a very nice alpha helices. But if you carefully look at it, some alpha helices are not in the right spot. It's shifted, although the helix itself is right. And if we combine these two together, then we have about 95% correct. AlphaFold 2 does 92%, but by combining tracing plus AlphaFold, we get a little bit more gain. So these are the two typical scenarios of advantage of tracing and advantage of AlphaFold. The method also works for multi-chains. Now this is some comparison between the deep main mast and some other methods. So if I compare deep main mast base, the tracing against AlphaFold 2 on this 178 cases, well, this is at the X and Y axis, a TM score. This is a kind of percentage of correctly aligned positions. Well, it's like it's a tie almost. 91 cases, tracing worked better. 87 cases, AlphaFold was better. It's clear when AlphaFold, when the deep main mast tracing wins and when AlphaFold wins. If the resolution is relatively high, 3 to 3.5, then tracing works better because I can see the structure. But if the local resolution is low, then well, tracing doesn't work, and then AlphaFold 2 wins. Now, but if I combine AlphaFold 2 and then tracing, base plus AlphaFold 2, the full pipeline, then this deep main must definitely win over AlphaFold 2 alone, because it can take the good part of both of them. Now here, I'm also comparing with some other methods, including Model Angelo. Model Angelo works very well for in a high resolution, relatively in high resolution, but when it comes to lower resolution, like a 4 to 5, 3.5 to 5, then deep main mast are definitely usually better. Because we focused on the modeling of lower resolution, because I thought that's where the people need some help from the modeling methods. So this is some typical scenarios at 3 angstrom. This is PDB author's model. This is model Angelo. this, well, looks not bad. This is deep main must. Well, it's also good. But when it comes to 4 angstrom or 4.3 angstrom, Michelangelo started to make very fragmented structures, cannot connect those fragments, but the deep main must do a better job in connecting those fragments. Okay, so that's about the protein modeling up to 5 angstrom using deep main must. And then this is CryoRead. This is the same approach, but now for DNA or RNA. Idea is basically the same. You have a CryoEM map of about 5 angstrom. Deep learning will detect sugar, phosphate, and base positions, and then connect. Then after that, those detected points are connected, and then sequences are assigned, and finally make the atomic detail model. Some examples. If only a part of the structure is RNA, in this case, there's a small RNA here in the middle. This CryoRead only built this the model of RNA or DNA because the deep learning will only detect DNA or RNA position, nucleotide positions, and protein regions are completely neglected. On the other hand, deep main must do the opposite. So if your structure have both protein and the DNA and RNA, then run both, and then it will do the modeling in corresponding positions. Now, okay, so this is about the model validation called DAQ score. So here, idea is the same as deep main must, but now we want to validate if your model fits well to the density. So now the input is a CryoEM map and your atomic detail model. And what this DAQ score does is it uses the same deep learning network and then predict the 20 different amino acid type at each positions and C alpha position and secondary structure at each position of each amino acid position, C alpha position of your model, and then compute the score. And well, I just quickly explained the meaning of this score. So suppose, okay, the residue position I in your model has tryptophan at certain position in your map. Then this numerator is a probability value of tryptophan at that position. That is P. And this is supposed to, if tryptophan is really correct, then you are supposed to have very high value for this probability of tryptophan at that position. And then this is normalized by that tryptophan probability computed at each atom position in the entire map. So it is normalizing it. And if the tryptophan is very correct, at least I would expect that tryptophan position probability of that position is at least better than overall tryptophan average or from the entire map. And if that is the case, then this fraction is larger than 1, and if you take a log of it, then it will be a positive score. So if you see the positive score, your amino acid assignment at that position is supported, but if it is negative, that amino acid is even worse than overall average of the map. So here, okay, so we validated in PDB entries from CryoEM map which have two versions of PDB structures. Often authors will make a correction to the structures that makes the second version or third version of the model. And we applied our score to the initial version and then second version. So this is one typical example. Here in this PDB entry, the two structures are very different. So here, the inconsistent regions between the first version and second version are shown in magenta. In this case, author even deposited the correction note to the journal. So the second model is probably very trustable. Even the author agreed author made lots of changes. And many changes made is actually the sequence shifts along this alpha helical regions. So which is shown in red here. So in this diagram, Okay, so x-axis is the position of the amino acid, y-axis is the DAQ score of the three different scores. So, all right, blue is amino acid type score. So amino acid type score are bad in those four regions. Those are the three alpha helices, and this in the top part. And then these three regions corresponds to these alpha helical regions. So these alpha helical regions have a low amino acid score, but if you look at this atom score, green, the score is not bad, positive, which means which this is a typical case, which indicates that C alpha position, helix structure itself are correct, but amino acid sequence assignment is shifted. So in this part, this is the first initial part, the top part, and for these regions, actually both the C alpha positions and the atom position, atom score, and amino acid type scores are low, which means that probably C alpha position also needs to be corrected. So okay, in summary, okay, if amino acid assignment is wrong, then the score distribution look like this. So it goes to negative. Distribution shifts to the negative. When the amino acid assignment is right, so this is a blue score distribution. And well, there's some overlap, but if you see the DAQ score lower than minus 0.5, for almost all the cases, the sequence assignment is wrong. And it's good to double check, how, if you could improve the modeling. We have a database, and we are monthly updating this database, and then this is, you can take a look at it. You can see the DAQ score assignment of the structures from CryoEM map. Now people started to use DAQ score. Well, compared to Q score, this is a common question we get. Q score tells you the like overlap of the volume and the resolution between the atom you placed and then density. Now the DAQ is a bit different. DAQ will use the deep learning methods, and the deep learning methods will look around the neighboring also the density patterns. So there are cases that amino acid seems to be fitting okay to the density, but actually you are fitting the wrong amino acids. So if the amino acids are fitting okay, but the assignment of the amino acid pattern is wrong, DAQ can really tell it. It is now included in PDBJ, the PDB branch in Japan. So from the CryoEM entries from PDBJ is linked to the DAQ score. I skipped this, but there is this tool is also available on our web server, and this is to fix once you identify the wrong region in the model, DAQ refine will automatically fix it. If the mistake is small, DAQ refine can fix it. But if the error is everywhere, well, that's over the scope of the DAQ refine. But yeah, well, please try it. Now, okay, so we have also the structure fitting methods, and this method works up to around 10 to 15 angstrom resolution. What it does is that you have a cryo-EM map and protein sequences, and then it does a structure fitting of AlphaFold models. But when the resolution is 8 to 10 angstrom, even fitting the structure fitting is not easy. So now the cryo-EM map is modified, like kind of sharpened, but not really sharpened. The main chain positions are emphasized by diffusion model. This is another deep learning methods. Now we know that the main chain position better, and by using that emphasized C alpha positions, we can do the structure fitting better. That's a diff modeler. Some examples, it can work also up to over 10 angstrom. Now, we also have a newer version of the diffusion model called DM Cloud. So DM Cloud will do the local structure fitting instead of global structure fitting. That we will submit this paper soon, but it is already available on our EM server. Now, okay, so here, this is my last slide from my side. And all of the tools are available on this web server. You can just click the methods you want to use, and then upload your map and then sequence, and then this server will do the modeling automatically. Of course, all these methods have source code made available on GitHub, and then some of the methods are also available in SPGrid, but I think that this server gives you the quick start because you don't need to do any installation. All right, so then I will pass this to Genki. So I stop sharing now.
Okay. Actually. Okay. Everyone, my name is Genki Terashis. Professor Kihara already explained us the detailed algorithm. So in my part, I will show how to use our program, especially the EM servers, and also I will show the GitHub page. So today I'm going to talk about these tools, 7 tools. So DeepMainMath is a de novo structure modeling tool, and CryoEM DNA RNA structure modeling, and DiffModeler using the diffusion model, enhancing the backbone positions, then fits the structure. And DiffModeler sequence is including the DiffModelers and the sequence search, structure search against the PDB database and AlphaFold database. DM Cloud is another new version of the DiffModeler using the diffusion models and using the point cloud matchings, then local fittings. DAQ is a model quality assessment tool. Then DAQ Refine is a model refinement tool. Okay, so first of all, our server, and the address is here, has a account system, so login system. Once you register your account, username, and email address, then you can keep your job history in our servers. So once you submit the job, then job is done, you will get some notifications from our server. Then also you can check the job history like this. Then you can easily explore that your previous job and models and results. So we strongly recommend to do the registrations. But of course, you don't have to. So you can submit a job, then wait for a few hours, then you just check the same web page again, you can see the results. Okay. So first, I will briefly show that DeepMainMath, servers and algorithm. So as Professor explained, that the DeepMainMath is a de novo protein structure modeling tools. So suppose you have a density map, then DeepMainMath use the deep learning method 3D unit, then identify the amino acid type and atom positions. They're using the big routing problem solver, connecting these predicted CR positions. They also assign the amino acid sequence, then build the entire full atom complex structure. This is a DeepMainMath. If you have AlphaFold model, AlphaFold 2 or 3, or some PDB related PDB structure, you can input the structure information as an additional input data. Then you can get the final 3D structure. Okay, this is a DeepMainMath. So DeepMainMath server is available in our EM server, suite servers. So this is the top page of our server. Then here, there's an algorithm tab. Then if you click here, then it's pulled down the all available programs. In the top left, there is a DeepMainMath link, and just click here. Then you can go to the job submission page. So address is here. The input file is a sequence file in the FASTA format and EMR. Then, as I said, if you have a structure information, AlphaFold models or other predicted model, you can input the data. So this is an exact example of the input data. So EMMAP is this EMMAP 1461. Then I use the AlphaFold models, but the domain orientation is slightly different, cannot fit well to the density map because AlphaFold 2 models have a different domain orientation. Therefore, this target, this is a homotrimer. So FASTA format sequence file has a 3 sequence data. These are input data. And this is a DeepMainMath job submission page. So you can use the control levels. So because the EMMAP has a lot of noise data and unrelated density informations, if you use the control levels, sufficient control levels, then you can reduce the computational time. But if you don't know the good control levels, you can put the 0.0. But in that case, our deep learning architecture, deep learning part is search against the all entire map regions. Here you can upload your EM map, CryoEM map, then upload the sequence file. Then this is the options. If AlphaFold2 structure or PDB structure is available, please click here, then upload the models. Then if you know that this DM map contains homo-oligomer structure, please turn on this option as yes. Then this is the full atom construction part. So because the DeepMainMath is tracing the backbone and building the structure, but side chain is not built by the DeepMainMath. So to build the side chains and define the side chain orientation, please turn on these Rosetta options. Then DeepMainMath use the Rosetta modeling pipeline. But if you want to know, if you just want to see the backbone tracing result or backbone overall shape, please turn off these options. Then you can get the result quicker. Okay, so this is the actual examples. So from the DeepMainMath result page, it will show the whole results. The first model is a backbone trace result. So C alpha model, it has only showing that the positions of the C alpha. The model 2 showing also the C alpha models, but it's only showing that the confident regions. So because the DeepMainMath must assign the confidence level at each C alpha positions, then if that position is not confident by the DeepMainMath, then model 2 doesn't show that low confident regions. So model 1 showing the entire traced backbone positions, the model 2 show only the confident regions. And the model 3, it shows the full atom structure. Then this model is colored by the DAQ score. So DAQ score is our deep learning based residual-wise quality score, the evaluation scores. So it's comparing with the predicted amino acid type and density pattern and predicted models. Then if this position is high DAQ score, high confidence region, it's colored by the blue. But if the position is not confident, it's colored by red. In this case, almost all regions are colored by blue, but some outside the loop regions, these positions are not confident. Then in the model 4, low confident regions are removed. It shows only the confident, high confident regions. Next tool is a CryoRead. CryoRead is a DNA RNA structure modeling tools. So from the density map, the deep learning method predicts the phosphate, sugar, and base positions. So from the input map, it automatically identifies that this position is a phosphate, this position is a sugar, then this position is a base. Then by using that information, it traces the backbone positions by connecting the phosphate and sugar positions. Then based on the predicted base information, then it assigns a sequence. Then finally, it automatically builds a flat structure. This is a CryoEM. A CryoEM is also available in our servers. So you can select the control level, and also you can specify the resolutions. It's used for the flat refinement part. Then of course you need to upload the EM map. Then you can upload the FASTA file. In the CryoEM, if you don't have a sequence information, then CryoEM automatically assign the sequence. So in that case, you don't have to put the FASTA format sequence file. Then after click the upload buttons, then wait for the 2 or 3 hours, you can get the result like this. So this is the input density map. Then it automatically identifies that the DNA, RNA regions, then build the full atom structure. So this is the CryoEM. Okay, then next modeling tool is a diff modeler. So diff modeler is using the diffusion models to enhance the backbone positions. Then using the traced backbone positions, it performs the rigid body fittings by our VESPR. So input density map are enhanced by the diffusion models. Then backbone position looks much sharper. Then from the sequence, so we can generate the AlphaFold models. Then predicted AlphaFold models are fitted into the enhanced backbone positions. Then finally, it can build the large entire structure. This is a diff model. Oh, yeah, okay, Professor already showed that this is the results. So by using the diffusion models, backbone position is clearly enhanced. Then we can build the complicated large macromolecule structure. I want to show the very interesting case. So this is a resolution 3.45. So diff modeler is originally designed for the lower resolution, 5 to 15 angstrom long. But also we noticed that our method is very useful for the higher resolutions. So this is AlphaFold models. This is the input map. Then diff modeler enhance the backbone positions. Then this is a backbone enhanced map. Then finally, it can generate the entire structure by fitting the AlphaFold model. If we carefully check the output files, so left is a input density map. Then it looks like the helical structure, but helical pitch is not clearly visible. But interestingly, by using the diffusion models, it clearly shows the helix pitch like this. Then by using this diffused map, enhanced map, the modeler can build a very accurate structure fitting. So this is a recently published paper in the Nature in the May this year. So our collaborator used this enhanced map to confirm that the backbone positions and also the kinks in the transmembrane regions as the GPCR. So diff modeler can build the atomic model automatically, and also the enhanced map is very helpful to check the local backbone structures and kinks of the helical structures. So diff modeler server is also available in our servers. So click the algorithm. Then here, the modeler link is here. I will show the actual examples of the input file. So this is the input file 6824 EMD. Then this is the input AlphaFold model. And this is a job submission page. So you can specify the control level here, then put the approximate resolution number, then input your EM map. Then here, you can upload the AlphaFold models. So if you have multiple AlphaFold models, please add the multiple your models here. So here you can upload the AlphaFold model, then also specify the number of the copies. If you have another AlphaFold model, please click this plus icon, then put the information, more additional AlphaFold model. It's very simple to use. And this is the result page. So you can show the input density map and also assembled fitted PDB structures. So in this page, you can also show that the PLDT values from the AlphaFold model and also the fitness scores. So it's comparing with the density map and assembled structures. Then if the fitted structure fitted very well, let's show the blue color. But it's not fitted well, it shows the red colors. We also have a modular sequence server. So in this server, if you don't have an AlphaFold model, but if you have a sequence, so you can use the sequence information as a query. So suppose you have a density map, low resolution density map, and also you have a sequence file. Then by using this sequence, our server search the most related protein structure from the AlphaFold database and also the PDB database. Then once our server finds the related protein structure, then it fits into the density map. This is a diff modeler sequence server. So if you don't have a structure, please try to use this diff modeler sequence server. The job submission page is also very simple. Specify the control levels, then approximate the resolutions, then put the EM map. Then for sequence information, please use the FASTA format file. So here, there's an example of the FASTA format files. So A, B, C, D, these 4 chains are this amino acid sequence, and EF chain of this sequence. Then please use the 2 separate sequences. So this is the input FASTA file. Then click the upload button. It automatically search against the AlphaFold database, PDB database, then assemble the macromolecules. Okay, then next tool is a DM Cloud. This is a new tool. So this is a point cloud-based local structure fittings. So we are preparing the papers, but server is already available. Please try to use this latest model fitting program. So link is here. So DM Cloud is using the diffusion models, but instead of using the density map, it converts to the point cloud. So density map is converted to the point cloud, and atomic model is also converted to the point cloud. Then using the computer vision architecture, then comparing the point cloud matching method. Then build the entire structure. So this is the DM Cloud. So this is the job submission page. You can specify the control level and EM map. Then DM Cloud have a new features. So you can visualize the shape of the density map. So changing the control levels, then click the here, you can visually check the control levels. Then, and this option is specified as this input PDB structure is AlphaFold based models or not. Then you can upload the PDB format file, protein structure models. If you have a multiple different structure, click the plus button icons, then upload the multiple structure model. Okay, this is an interesting example. This rainbow colored structure is a PDB deposit structure, PDB8 DTM. But AlphaFold 2 generates a completely different structure. So some part of the structure looks well fitted, but entire shape and domain orientation is completely different. But by using this magenta AlphaFold 2 structure, it automatically identifies the corresponding position using the point cloud local feature, then assemble, then fit into the density map. It's the local structure fitting method. They're also fixing the orientation of the domain. Okay, the next tool is a DAQ score, deep learning amino acid-wise model quality score. So using the same architecture in the deep main mask, machine learning method can predict the probability values of the amino acid type and atom type and the secondary structure type. Then by comparing that probability values and also the amino acid type in the PDB structure, then DAQ assign the model quality score. If the predicted amino acid and models have the same amino acid type, then it's better than the average of the predicted values among the all other regions, then it has a high score. But it's not consistent, then it shows the low quality scores. The low quality score regions are colored by red, then high quality regions are colored by blue. So DAQ score is a very useful to identify that the long sequence assignment regions. So this is a very interesting example, 6L54 and 6Z3R. These two PDB entries have an exact same sequence, and shape looks very similar, backbone position looks very similar. But interestingly, the same region number 425 and 423 are modeled in a different place. So 6L54 modeled here, here, but these pink structures about 425 modeled here and 423 modeled here. Even in the PDB database, these two structures are inconsistent. But using the DAQ score, the DAQ identified as that, okay, these regions have a negative DAQ score. It's highly possible that this left PDB structure is not matched well with the density. So this is a DAQ score. So DAQ score server is available in our server. Click algorithms, then here, click DAQ score. You can just upload the PDB file and CryoEM. That's it. Then it automatically evaluates the input atomic models. I will quickly show the recent examples, 31563s and PDB entry, the 7FET. So this is an output page. So it has a 3 chains, A, B, C chains. Then it shows the 3D atomic structures colored by the DAQ score. DAQ-AA is the amino acid assignment score. Then x-axis is the positions of the PDB structures. The y-axis is a DAQ score. So if the DAQ-AA score is lower than 0.5, minus 0.5, it's highly possible that these positions are wrongly modeled. So here, this position is corresponding to this beta hairpin structure. Okay. Then server also show the C alpha DAQ C alpha score. So C alpha operation is correct or not. So this case, these positions are negative 0.5. So it's corresponding to these regions because this region is resolution is not good, and also there's some missing residue, missing positions, missing C alpha positions. Then DAQ said it is highly possible that's the wrong model. Okay, then this is the final tool, so DAQ refine. So DAQ refine is an automated model refinement tools. So you can input the EM map and the PDB structure. Then it automatically found that the pitch position is a low score, low quality score, DAQ score. Then by using the AlphaFold, customize the AlphaFold tool, it builds the atomic models, then try to improve the low DAQ regions, low DAQ score regions. So input structures about low DAQ regions are a lot of places, but using the DAQ define, it automatically defines the structure. Then overall looks very nice. So all regions have a positive DAQ score. So you can input the structure, models, and density map, then approximate resolutions, then DAQ score cutoff. If you use a 0.0, so that means if DAQ score is lower than 0.0, then AlphaFold and Rosetta try to improve that region. This is the examples. So model 1 show the input PDB structure. Then DAQ score said, okay, this helix has a low DAQ scores, so maybe inaccurate. Then model 2 show the after refinement, refined structures. So here, so all these helix regions are colored by blue, so that means that these regions are improved and has a positive DAQ score. Okay, so I have 5 minutes, so I want to show the our server quickly. Okay. Okay, can you show the web page? Okay. Okay, so this is our website. Then here is the algorithm tab. Click here. Then you can jump to the algorithm, so programs. So here I want to show the DAQ score. Here, click here. Then it has a clear instructions how to use, then how we can interpret the result. And here, then you can upload the PDB structure. For this case, I want to try this A7 FET. Click here, then upload. Then CryoEM here, upload. Then click here. Then now the server is transferring the file to the system. Yes, then our job is starting. So here is the job manager page. Then my job, I can confirm that this is my job. Then now the DAQ squad job is running. I already used submitted a lot of DAQ squad jobs, but now it's running. Okay, so then I can show the result page. So this is the result page, so DAQ score. So 30 minutes ago, I submitted the same job. Then this is a DAQ AA score, colored by DAQ AA score. So we can see that these regions, these data hairpin regions have a very clear red color. So that means that DAQ says that this region might be highly possible that's a long sequence assignment. And also there is a 2D plot. We can see that these regions have a very low negative DAQ score. Okay, so yeah, it's very easy to use. So please try to use our web server. Then also the source code, all source code are available in our GitHub page. So yeah, please access to the GitHub Kihara lab, this page. So this page, we released all deep learning architecture, the weight and script. So these are individual sites, but each site has a clear instructions how to install the code and how to manage, how to process, like so on. So for example, this is a GitHub page of the DAQ score, and this is a diff modeler code page. So please visit to our GitHub page. Okay, this is the summaries. So I explained the 7 tools. So deep mass query, diff modeler, DM cloud, DAQ, and DAQ defines. So we are providing the models, de novo structure modelings, DNA RNA structure modelings, and diffusion models, then enhancing the backbone positions, and also the model quality assessment tools and model refinement tools. All tools are available in our servers, so please try to use our tools. And please give us the feedback. Okay, this is the end of my presentation. We appreciate our lab member. I appreciate lab members, especially the Professor Kihara. Thank you for your attention.
Wonderful. Thank you, Genki and Daisuke. Yeah, really great overview of all the so many tools that are coming out of your lab. It's fantastic to see. And I just want to emphasize how, wonderful it is to have access to these tools on a web server. It really does help accessibility, and the server you guys have put together is really beautiful and user-friendly. So it's fantastic. I just want to note, Daisuke has really been busy addressing many of the questions posted by the audience along the way. Those should all be recorded on the answered questions. So if some of those questions resonated with you, please go look at Daisuke's responses. I do have some, maybe a couple broader questions that I could maybe throw out. I noticed, so this may relate a little bit to some of the questions that come up during Mark's talk, just kind of fundamental questions about these AI tools, why they work well for some and why they don't work so well for other components of biomolecules. So you developed two separate tools, one for protein and one for nucleic acid. Does that reflect kind of the limitation of having training sets that have both types of molecules in one platform? Maybe you could comment on that.
Yeah, I think, yeah, so yes, that's one reason. And also there's a historical reason. We started started on protein modeling first, and then realized that we can also do the DNA RNA structure modeling. That is a historical reason. But now we are addressing well, although we haven't posted the methods yet, we have a tool which does both altogether. So currently in this server, web server, you can choose complex modeler, in which does the protein modeling and DNA modeling, and then do the combine the model together. So you can do that on this server, but we are also developing it. But yeah, you're right. Mainly, well, it's coming from the data set limitation and also historical reason.
Gotcha, thanks. And then I'm just curious too, is it really that we're just limited in our data set representation of some of these smaller or more unique like ligands, solvents, ions for training these models? Or is there really something more fundamental to these AI enhancement methods that lead to inconsistent results for some of the things that Mark highlighted in the previous talk.
So okay, you asked the ligands, right? So ligand modeling, yeah, we are also actually in the process of developing the ligand modeling methods. But ligand modeling is more difficult. Actually, the data set is even doubtful sometimes, because if you look at the PDB entry, and you see the ligands are not really fitting to the map, and or sometimes, well, authors do not model all of the ligands, especially if the ligands are the general ligands like NAG or some like small sugar molecules all over the place. Then that's makes our work difficult because I don't have a ground truth. It's clear that
Right.
some of the density needs to be filled with ligands, but also didn't do it because it's probably less relevant to the biological questions. So that's another difficulty for like tool builders like us.
Gotcha. No, that makes sense. Thanks. One last question I noted, and it came up on this last slide here. You do give some guidance on size limitations. Is that just for the server, or are those different for the standalone software versions?
Oh, okay. So yeah, size limitations is that if we use the Rosettas or AlphaFold, so it has a size limitations for the GPU memory size. So this is the main reasons. But if you have a
Okay.
80 GB large memories, then yeah, user can enhance the size limitations.
Okay. Cool. And is that like are there any other use cases where using the standalone versions are recommended over the server versions?
Okay, yeah, there are several reasons. So for example, if user do not want to submit own data, some CryoEM data, but want to compute the deep main mask, then in that case, the standalone version is one of the choice.
Great.
But I guess Genki, so in terms of the like a size limitation or quality of the model, is there any difference between server and standalone program?
Basically, no.
Yeah, I think it should be the same. But then I
Understand.
I noticed that there was one question from on the question answer page about the difference between the server, CryoRead server, and the standalone program. And I'm not sure why it happened. And well, I encourage them to contact me. We will check what happened.
Gotcha.
Good.
All right. Well, we'll give people a little bit of a break here before our next speaker. But thank you both again for a wonderful talk.
Thank you very much.
Thank you.